Hello guys, welcome to our channel Ask Prep. Today's series is for 11th class biology. We will be studying chapter 13, photosynthesis in higher plants in detail. We have also added important notes for your exam preparation. All the best for your exam. Starting with photosynthesis. Overview: photosynthesis is the process by which green plants convert carbon dioxide, CO2, and water, H2O, into carbohydrates, C6H12O6, using sunlight. This process transforms solar energy into chemical energy stored in glucose, sustaining life on Earth by providing food and oxygen for living organisms. Key components of photosynthesis, or what we know about photosynthesis. The natural physico-chemical process of photosynthesis is essential to life as we know it on Earth. The most significant biological process required for human existence is photosynthesis. Sunlight. Light energy from the sun is the driving force of photosynthesis. Chlorophyll. The green pigment in plants that absorbs light found in chloroplasts. Carbon dioxide, CO2. Absorbed from the atmosphere and used in the synthesis of carbohydrates. Water, H2O. Taken up by roots from the soil, it plays a critical role in producing oxygen as a byproduct. Photosynthesis. Reaction. The overall chemical equation for photosynthesis is 6CO2 plus 6 H2O plus light C6H12O6 plus 6O2. Next is experiments demonstrating photosynthesis. Variegated leaf experiment. This experiment shows that chlorophyll is essential for photosynthesis. Variegated leaves, which have both green and white parts, produce starch only in green areas because they contain chlorophyll, confirming its role in photosynthesis. Light screen experiment demonstrates the importance of light in photosynthesis. A part of a leaf is covered with a light screen, black paper, and after exposure to sunlight, only the uncovered portion produces starch, proving that light is necessary for photosynthesis. Moles experiment. This experiment demonstrates that carbon dioxide is necessary for photosynthesis. Leaves are partially exposed to an environment without CO2 using potassium hydroxide, which absorbs CO2. And only the exposed parts to CO2 produce starch, confirming the role of carbon dioxide. Let's understand in detail.
With the help of a variegated leaf experiment, we can prove that chlorophyll is essential for photosynthesis. To do this experiment, we need a potted plant with variegated leaves. We have to keep the plant in dark for 3 days. Do you know why? So that all the starch present in the plant gets used up. Now place this plant in sunlight for about 6 hours. Now detach a leaf from the plant and mark the green areas of it. Now place the leaf and its green borders on a butter paper. Next, place the leaf in boiling water for a few minutes. Take it out and place it in a beaker containing alcohol. Now place the beaker with alcohol into a water bath. Heat the setup till the alcohol begins to boil. The leaf loses its color. The alcohol turns into green color. Now take the leaf out and dip it in a dilute iodine solution for few minutes. Take out the leaf and rinse it. Observe the blue colored part of the leaf and compare it with the paper tracing. Here we observe that starch is prepared only in the green color parts of the leaf. Hence we prove that chlorophyll is important for photosynthesis. How can we prove that carbon dioxide is necessary for photosynthesis? Write an experiment to show that carbon dioxide is essential for photosynthesis. we can prove that carbon dioxide is necessary for photosynthesis by a simple experiment. To do this experiment, we need two healthy potted plants of same size. We have to keep them in a dark room for 3 days to detach them. Place the plants on glass plates or smooth finish tiles. Place a watch glass containing potassium hydroxide by the side of one of the plant. Cover both the plants with separate bell jars. Why to place the plants on glass plates? Because we want the plant to be in a airtight environment. So we have to take a smooth surface at the bottom and a bell jar at the top to keep it airtight. Even then some air can enter inside through the edges. How do we stop it? That is why we have to apply Vaseline or grease at the edges to prevent the entry of air. But still there is some air in the bell jar, that means there is some carbon dioxide inside it. How do we remove it? That is why we have placed a watch glass with potassium hydroxide which will absorb the carbon dioxide present in the jar. So now our setup is ready. One plant without CO2 and another plant with some CO2. Let the plant stand in sunlight for 2 hours. Now pluck a leaf from each plant and test them for the presence of starch. Do both the leaves show the presence of same amount of starch? No. The plant with KOH plant plate shows no signs of starch, but the plant without potassium hydroxide could prepare some starch. By this experiment, we can conclude that carbon dioxide is essential for photosynthesis.
Historical experiments. Joseph Priestley, 1774, discovered that plants purify the air by absorbing phlogiston, CO2, and releasing oxygen. He proved this by showing that a plant can keep a candle burning and a rat alive in a closed system. Jan Ingenhousz showed that sunlight is required for plants to produce oxygen. He noted that plants release oxygen in light but absorb CO2 in the dark. Julius von Sachs, 1854, discovered that plants store glucose as starch. T.W. Engelmann used algae to discover that photosynthesis is most efficient in blue and red light. See the image. Next is, where does photosynthesis take place? Green plant cells contain chloroplasts, a semi-autonomous endosymbiont cell organelle with 3 membrane systems. It is composed of 3 primary components: a thylakoid, a matrix, and an envelope. An envelope is made up of 2 membranes, an inner membrane and an outer membrane. Periplasmic space is the region between the outer and inner membranes. Matrix, double-stranded circular DNA, the 70S ribosome, the Rubisco carboxylation enzyme, the PEP carboxylase enzyme, magnesium, Ci, and other elements are found in a semi-liquid substrate known as a matrix. Pigments involved in photosynthesis: photosynthetic unit, PSU, light harvesting complex, LHC, quantasome. The units of photosynthesis, known as quantasomes, were identified by Roderick B. Park. They are found in the chloroplast's thylakoid membrane and are made of lipids and proteins. When light is converted into chemical energy, this is the smallest group of pigment molecules involved. Their reaction, photo, or trap centers are always made up of a single chlorophyll molecule that consumes pigments that absorb light. Photosynthetic pigments. 3 main types of photosynthetic pigments are as follows. Chlorophyll, the primary pigment responsible for capturing light energy. Carotenoids, accessory pigments that help capture light, especially in the blue and violet regions. Phycobilins, pigments found in some algae, absorbing different wavelengths of light. Photorespiration. In C3 plants, photorespiration occurs when oxygen binds to rubisco, leading to the production of phosphoglycolate instead of phosphoglycerate. This process produces no useful energy, no ATP or NADPH. C4 plants, however, have evolved mechanisms to minimize photorespiration by increasing intracellular CO2 concentrations. Absorption and action spectrum. Absorption spectrum displays which wavelengths of light are absorbed by pigments. Example, chlorophyll absorbs blue and red light. Action spectrum shows the rate of photosynthesis at different wavelengths with peaks in the blue and red regions of the light spectrum. Light reaction. T.W. Engelmann created the first photosynthetic activity spectrum in 1882. The visible spectrum's red light is when photosynthesis is at its fastest. The visible spectrum's blue and red sections are where photosynthesis mostly takes place. Other wavelengths do not see the same rate of photosynthesis because the accessory pigments in these regions are limited to absorbing light energy and transferring it to chlorophyll A. Only in chlorophyll A reaction center does the light reaction take place. The process of photosynthesis involves the oxidation of water and reduction of carbon dioxide through a redox reaction. Non-cyclic photophosphorylation. Another name for it is Zeitz scheme. It is made up of PS1 and PS2. Chlorophyll A 660, 673, 690, chlorophyll B, chlorophyll C, or chlorophyll D, carotenoids, and phycobilins make up photosystem 2 PS2. Phytoplankton, a 680 is the response center. In stromal lamellae, it occurs. NADP plus takes the electron instead of it going back to the reaction center. When water is photolyzed, electrons are used, which results in the production of NADPH2 and ATP. Advantages of C4 pathway. Because PP has a stronger affinity for carbon dioxide, the C4 route is more effective at removing carbon dioxide from the atmosphere and does not include photorespiration. Due to the bundle sheath cell's proximity to the water supply surface, the impact of water stress on C4 plants is reduced. Because it contains organic acids, it can withstand stress. C4 plants fix the same amount of carbon dioxide in order to minimize water loss. In terms of carbon dioxide fixation, C4 plants were twice as effective as C3 plants. Thanks for watching the video. We will drop the notes in the description of this video. If you found this video helpful, please like, subscribe, and share. See you in the next video, and all the best for your exam.